So what I'm going to focus on today, three key words in the title: Bayesian adaptive design. And Bayesian meaning one gets as inference at the end of the trial is based on a posterior probability of a hypothesis being true, an alternative hypothesis, given the data, which is a little different than what one would traditionally make inference using a p-value, where a p-value is the probability of data given a hypothesis. What the Bayesian posterior probability allows one to do is to interpret the probability, again, of a hypothesis for this particular trial at the end of it. The adaptive design part allows for flexibilities to take place during the trial, during the conduction of the trial, where it can move to different places, so you can optimize finding the answer. But a key feature of the word adaptive design, as defined by the US FDA, maybe about 10 years ago, was the first guidelines to come out about Bayesian adaptive designs. And this has been defined before, but the key to it is a pre-specification of the rules of how the adaptation takes place. And one practical implication of that is it allows one to calculate trial operating characteristics, such as type 1 error rate, which is really, really important to regulators and IRBs and human subjects committees. And then allows you to calculate power and other operating characteristics. And because they're so flexible, meaning the adaptive design is so flexible, we have to, the current state of the practice is to calculate operating characteristics using simulation. So I'm going to talk about this idea in this particular trial design that I'll present today, as Simone kindly introduced, about using the primary and secondary outcomes. Traditionally, adaptive designs are driven by a single primary outcome, and we're going to bring in the secondary outcome into the trial, in the primary trial design. So to give a little bit of a background here, the drug we're talking about is TWO-ENDPOINT, which is 20 milligrams of dexaphene and 0.45 milligrams of conjugated estrogens. And it's FDA approved, federally approved for treating menopausal symptoms and preventing osteoporosis in menopausal women. So that's what it's indicated for. A group here is interested in breast cancer prevention, led by Dr. Carol Fabian. And we did a pilot study in which we had 28 women who were at increased risk for breast cancer. And we showed an improvement in biomarkers, fibroglandular volume, FGV, and benign breast tissue proliferation, Ki-67. And because of this improvement in this single arm design, it is hypothesized that this could be potentially indicated for breast cancer prevention, in addition to treating menopausal symptoms. So hence the next trial design, the phase 2B trial design. So I'm going to talk about today, I'm going to talk about the study design and some of the innovative features we've added to this trial design, the statistical model, and then, as I alluded to in the introduction, we're going to show simulation results, meaning we're going to use computer-generated clinical trials, a bunch of them, to show how the design behaves relative to other kind of trial designs, to get a sense of what kind of improvements we get. And then, as we always do before we actually conduct a clinical trial, we take a single data set, we create a single data set, and we do a mock interim analysis. That is, we have the team, the clinical team input kind of mock data into our clinical trial management system. And from that, all the way to the end, we do a mock interim analysis to make sure that we're okay. Because one of the things about the adaptive design is you're doing interim analyses as you go along. It's almost like mini final analyses along the life of the trial. So the primary objective, we have we would like to determine at 6 months post randomization if BZA plus CE significantly reduces fibroglandular volume when compared to controls, right? The pilot study was done only on BZA plus CE. Now we're bringing in a control, and you can think of randomizing the participants to one of the two arms. The secondary objective is to see if Ki-67 is dropped when compared to control. And this is only among participants that have a baseline Ki-67 greater than or equal to 1%. So it's potentially a subset. And then we have tertiary and exploratory objectives with other risk and mechanistic biomarkers. So how do we tackle this problem? We have this primary endpoint and we have this secondary endpoint that we want to highlight in the trial design. So one could rank order the importance of the endpoints. These are ways in which multiple endpoints can be handled in a trial. And you can think of allocating, you have certain costs in the trial associated with type 1 error. A lot of it could be allocated towards the primary endpoint and a little bit less to the secondary endpoint. And that could be done by rank ordering them. We could treat both endpoints as being equally important, and we could apply a Bonferroni correction. Our approach is to test the success of a single primary endpoint at the end of the trial. We're interested in FGV primarily once we're done with the trial, whenever that is. But we stop the trial only if we have adequate information for the secondary endpoint also. So you can see that if you're familiar with these kind of trial designs where you're allowed to stop early for a big signal, if you only use the primary endpoint to do that, then you might not have enough information for that secondary endpoint. You might regret that at the end of the trial. So here is the schedule of events. You have the patient comes in, is consented, then becomes a participant, and they get a baseline visit. And then they're randomized to BZA plus CE or a waitlist control. So the waitlist control gets nothing, and then they have a 6-month study visit. The BZA plus CE, you wait for 6 months after taking the medication for 6 months, and they have a 6-month visit. And they become off study, but the waitlist control actually get the medication after that. And this is optional for the participants. And this whole part after the 6 months is optional in terms of the study visits and stuff. The primary is in this circle around 6 months. And the waitlist control seems is the right thing to do, given that it's indicated for other reasons, that the drug is indicated for other reasons. And at the 6-month visit, that is where we get another mammogram. So we have a baseline mammogram, a blood draw at baseline, a mammogram at 6 months, and a blood draw to get the primary and secondary endpoints. What I didn't mention earlier is the randomization is 1-to-1, so it's equally randomized across the two groups, but it's stratified by site. This is a multi-site trial. So here I am in USA at University of Kansas Medical Center. We're a site. And then we also have some sites out in California, and we have some sites out in Massachusetts. and we have a site in Chicago, Illinois. So it's stratified by site, and then we're also stratified by baseline high versus low FGV, as well as high versus low Ki-67. So this is what we're going to call our design, our adaptive design. We'll call a two-endpoint adaptive design because it has a primary endpoint and a secondary endpoint that drives the adaptive design. The minimum sample size of the trial is 60. So once we have completed follow-ups for 60 participants, and this is a potential follow-up because some of them may have dropped out, but basically 6 months after their baseline visit, we will perform an interim analysis, okay? And then there's a decision about whether to stop the trial at that point or to continue all the way to 120. So this has one interim analysis. And so the maximum sample size is 120. So then the stopping rules are a couple of different stopping rules here. It depends on the posterior probability that the reduction in FGV and the reduction in Ki-67, baseline to 6 month, in the intervention group is higher than the control. So that posterior probability is the probability of the alternative hypothesis given the data, okay? So we calculate both of them for both FGV and for Ki-67. We end up with two posterior probabilities. If both of those posterior probabilities are smaller than 0.5, that is, there's a less than 50% chance that FGV is better in the intervention group than control, and Ki-67 that's low, and we'll stop and we'll say there's a very low chance that this could be a successful trial. So this can save money, time, resources. Now, a success stopping rule would be if those probabilities, both of them are bigger than 0.9847, we have a really, really, really good chance of success. And, you know, we've enrolled 60 with completed follow-ups. We'll have enrolled some after that, possibly, and let's say 1 or 2, then we would follow up and recalculate the posterior probability, but only for FGV. If that posterior probability stays at 0.95175 or more, we have a successful trial, and we could say that the drug is indicated for reducing biomarkers for breast cancer. And so you can see here that the trial could stop either at 60 or at 120 participants enrolled into the trial. So what's the statistical model? Well, what we have is the biomarkers, FGV, the change in biomarkers, FGV and Ki-67, both of them have a normal distribution. So you can think of a two-sample t-test in this case. But we have a Bayesian version of the two-sample t-test rather than a frequentist p-value. And so attached to the parameters in this normal distribution are prior distributions. And so Bayesians will take prior information and update it with data to get a posterior distribution. The prior gives information to the trial as well as the data. But in this case, our priors are really, really not adding a lot of information. They're thought of as being fairly non-informative or weakly informative prior distributions. So they don't add a lot of information to the trial results. Most of the trial information comes from the data collected. Okay, so now we have our statistical model, which allows us to calculate posterior probabilities. There are closed form solutions to get Bayesian p-values from this. If you've ever studied a book, classic Bayesian book by Andrew Gelman et al., And there are some great books and methodologies all over the world. Best et al., who are the bugs developers, have some great books and great software. But there is a closed form solution to calculating this, but we use Markov chain Monte Carlo, MCMC, to calculate in general the posterior probabilities, and we do so in this case. I will say that in the trial design, as I mentioned before, how many participants you have in the final analysis is dictated by how fast you enroll participants. So we have a distribution that we make assumption about how we enroll participants. And we have a Poisson distribution that says, on average, we recruit 0.676 participants per week. And this is indicated, or this is known from our pilot study about how fast we were able to enroll them. And we'll assume an overall dropout rate of 10% in the study. So this allows us to do, as I mentioned, calculating the operating characteristics of the trial. So just to review, we have an early stopping for success, very stringent rule, 0.9847 in both. If you want to translate that 0.9847 to look like a p-value, take 1 minus that, it would be a really, really small value. Early stopping for futility is if both of those FGV and Ki67 posterior probabilities are less than 0.5, and then the final success is less stringent, 0.9517. Okay, so we have 2 endpoints for stopping the trial, but only 1 endpoint for final success. And the reason the interim analyses is more stringent with both here is to make sure that our secondary endpoint has enough information, so we don't regret not having information for that in addition to the FGV. So now let me talk about simulations. So this is where we're going to calculate the operating characteristics of the trial. The type 1 error, power, the average sample size of the trial design that produces the trial design from the trial design, and then how long it takes to conduct the trial. And I point you to this table at the bottom of slide 16, where we have assumptions about the different average drop in FGV and Ki67. And so this first row is none. You can think of this as being the null hypothesis, HO. So that the drop is the same in FGV, 2.5, and also in Ki67 is around 0. So what we're going to do is create simulated data from this hypothesis. And we will run through at each clinical trial the actual design. And we'll calculate posterior probabilities, and we'll determine for that trial, did we stop early? Did we find success? And we'll tally all of that in a row. Do it again, put it in a row, do it again, put it in a row. And you can do this 100,000 times is generally the recommendation. And then we calculate under the null hypothesis, how many times were we falsely saying there was an indication or success? And that's our type 1 error. Then to calculate power, we do the same thing under the expected hypothesis. This is row 2. You can see there's a bigger drop in BZA plus CE, negative 20.1. That was fed to us from the pilot study. Same with the Ki67. So then when we do this again, we do that computer experiments, tally everything, and we get power. We also get how, on average, how many times did we stop early? What was the size of the trials on average? We have a large effect. And then we also have a case where actually BZA plus CE is worse. This will really give us a good idea how well our futility rule works in stopping the trial. Okay, so now we're ready to move forward with the simulations. Okay, so this table is going to be broken down into pieces. I will say there are three different designs we considered. The first is the two endpoint adaptive. That's the design we've been talking about the last several minutes. I also wanted to compare it to a one endpoint fixed design. What that means basically is randomizing 1-to-1, enrolling 120 participants, getting their follow-up, and calculating a p-value of 0.0 less than or equal to 0.05. We do a Bayesian version of that, but that's the fixed trial design. There's no stopping early. The one endpoint adaptive just uses FGV to stop the trial early. Okay, so three comparisons. We want to see how well our two endpoint adaptive improves or is a compromise of the one endpoint fixed and the one endpoint adaptive. So the type 1 error is well controlled for all three of the designs. We sometimes say you want to compare apples to apples, and this says that we can. There's a little bit of a simulation error, but under this column here, it says power. That's essentially the type 1 error. They're all less than or equal to 0.05. Okay. So that says that we have unbiased designs. All designs also have approximately the same power. So this is under the expected effect, the large effect. Under the expected effect, all three designs have about 90% power. Not really a big difference. Under the large effect, all three designs have about 99%. Now, let's talk about how long these trials take. How many participants are enrolled in them on average? Well, in the no effect case, the one endpoint fix, you're never stopping early. So you're always going to 120. It takes about 184 weeks on average, but you get an improvement in that you get average number of subjects enrolled, 110, and taking about 13 to 14 weeks less for the one endpoint adaptive and two endpoint adaptive. They're really not different from each other, but you do see a nice improvement there. This is done because they're stopping for futility. Under the expected case, same thing, 120, 184 weeks. But the one endpoint adaptive actually reduces the average sample size by 14 subjects and 19 weeks less to do the trial on average. Whereas the two adaptive really looks like a one endpoint fixed. Really aren't getting a lot of savings there. On average, these trials are enrolling more to get more information about the secondary endpoint Ki-67. However, when you get a large effect, again, the one endpoint fixed, it looks just like it always has. It's a fixed design. The one endpoint adaptive and two endpoints, you have about a nice reduction, about the same improvement under the large effect. Under the worse effect here, this is because, again, we get improvement in sample size and time because there's a chance of futility. Okay, so in summary, what this table says is that there's improvements in going adaptive in that you get faster trials, smaller trials, about the same power. But the one endpoint adaptive has a little bit of a different feel than the two endpoint. It's probably a little easier to look at when you look at a figure. And so let's orientate ourselves to this figure, these four figures. In the first column, this has to do with the average sample size on the vertical axis. On the horizontal axis, it's the true drop in Ki-67. So you go from a big effect size to a smaller effect size. And this upper left quadrant is for large FGV. And you can see that as the endpoint is really Ki-67 has a large effect, that the two endpoint adaptive is smaller in size like the one endpoint adaptive. But as the true drop in Ki-67 gets smaller towards zero, it starts to look like a one endpoint fix. That's because the trial will keep going so that we get more information for Ki-67. And that trend also continues in the very large FGV. We get the same thing. Now, a lot of times, since we're not making a trial success based on Ki-67, we're most interested in the length of the credible interval. And that's a function, excuse me, of the posterior standard deviation of difference in Ki-67. You can see that the two adaptive endpoint starts to get smaller and smaller as we get true drop of Ki-67 is also smaller. Starts to look, so it's a similar compromise to that of the sample size. So the one endpoint adaptive requires a smaller sample size, but gives us less information about the secondary endpoint. Okay, mock interim analysis. So as I mentioned in the introduction, what our team does is once we have our final protocol set, once we have all of our case report forms built and placed on our clinical trials management system, we've tested the clinical trials management system, we've trained our clinical investigative team to use the clinical trials management system. We all agree that it's finalized. Then we ask the team to put in fake data for participants to test the system. And in that step, we also run through an interim analysis. And this helps us think through what the report will look like for the interim analysis. So the way the flow works, Velos is our vendor for our clinical trials management system. And this is an FDA certified clinical trials management system that we use. We certified in our department. Data from there are pulled to the shared drive, we call it S drive. And then from that, we use SAS and R to create a patient's mock report. And then the patient's mock report is called in by a program called FACS. FACS is a fixed and adaptive clinical trials simulation. And it is the state-of-the-art software program globally for doing these kind of designs. It's very, very flexible. The FDA respects the software program. You can design it, simulate from this software, and you can also run analyses in the software. And that's what we're doing here. We're pulling the mock data into FACS. And then FACS, we're running the analysis of where our design was took place. And then we're pulling a report in SAS and R, and then filling that into an interim analysis report. This is done in Markdown, so we can have as much reproducibility as possible in this data flow. And then that interim analysis report is in a Word document for all of the appropriate people to read. Now, we all read this one. This one's a mock one, but you know, in a blinded study, the statistical team would focus on the interim analysis report until it's appropriate to share with all investigators. So here's what the patient data file looks like in FACS. We have subject ID, region ID. Some of these aren't actually used directly. It's more of like documents of things, but things that are used directly in the program, in the analysis, is this dose index, the last visit, whether it's 0 or 1, baseline or follow-up, a dropout indicator, whether the participant dropped out or not. We don't have a baseline that's used in the analysis. It's just the difference in FGV. Then we have a response, okay, for endpoint 1, and we also have a response for endpoint 2, okay? So this is what is produced from FACS. Important outputs and facts is this weeks file. This gives you posterior summaries for all parameters, including posterior probability. We also get input parameters, a number of Markov chain Monte Carlo samples, and then also a burn-in. So we have an MCMC file. This MCMC file, which gives you posterior draws. If you have 1,000,000 draws, you'll have 1,000,000 rows. That's used to calculate credible intervals. So here's a mock interim analysis. Here are the results. We have estimates and 95% credible intervals for all parameters, including mean drop in FGV for both groups, its standard deviation, the difference, and then the most important thing for an interim analysis is the posterior probability of the alternative hypothesis, given the data, probability of posterior, the hypothesis given the data is 0.5979. So it's not less than 0.5, and it's not bigger than 0.98457, I think it was. And then the same thing for Ki67, the most relevant piece here is this 0.5118 posterior probability, which that didn't meet the criteria either, 0.9847. So none of them are met. So in this mock interim analysis, the pre-specified plan is to continue enrolling all the way to 120. Okay. So the calculation of credible intervals, you generate an MCMC file, you run the analysis, you get R and SAS pulled out, and you calculate percentiles 2.5 and 97.5%. You can also calculate different methods for credible intervals, including HPD is be appropriate, but this model tends to result in fairly normal posterior distributions. And so here's the proc import you could do in SAS, and then a proc univariate to calculate the percentiles in SAS. One thing we like to do is, because we're using Markov chain Monte Carlo, we'd like to get an idea of what the MCMC error is, because we're not doing this an infinite amount of times. This meaning posterior draws from Markov chain Monte Carlo. Okay, so what one does is change the seed, rerun the analysis, and get a posterior distribution. The seed meaning where you begin your random number generator. Okay, knowing the seed can reproduce the same simulation results. So if we change that, get replications of that, we will have posterior probabilities 10 times. Then we can calculate the standard deviation of those 10 times, getting a standard error of the parameter of interest. So this can tell us how many digits we can report the posterior probability in a report and be confident. So here is a result for the mock interim analysis. And basically, if you look down at the bottom, here's the posterior probability. We did 1,000,000 draws after 10,000 burn-ins. You get different posterior probabilities. And this MCMC standard error is 0.000276. Looks like we can feel very comfortable going up to 3 digits. If we wanted to go to 4 digits, we want to, increase the number of draws of the MCMC. So this is a really, really important step to understand what the MCMC error is. Those of you that are bugs users know that bugs does use a similar technique to get their MCMC error. So where are we now? We've started the trial. We started enrollment on December 14th, 2021. So it's been 17.4 months since we started enrollment. As of May 29th, there are currently 28 participants enrolled on the study. All of them come from our institution, KUMC, University of Kansas Medical Center, and 15 participants have completed this study. Other sites are onboarded and ready to enroll patients to become participants. We still have yet to have our second site enroll participants. And the design paper has appeared in trials that this paper is based, this talk is based on. And so here we have KUMC. What we have is part of our clinical trials management system is we give a report every Monday of where we are enrolled. We show plots like this. We also show posterior predictive probabilities, posterior predictive distributions of future enrollment by our target. And I don't have a plot for that, but we do provide that also. We have some Bayesian accrual prediction models that we use. So before closing, I'd like to mention a few things. Some of the co-authors we have from our team, we have Kimler, Kessler, Mudaranthankam, Young, and Fabian. Kate Young just defended her dissertation in January, so now she's transitioning from a statistician 2 position to a faculty position here coming up by the 12th of June. Berry Consultants, I'd like to thank them for the fax software, the license that we have at the University of Kansas Medical Center. Kate, Dr. Young, for building the slides today. And this work is supported by the National Institutes of Health and National Cancer Institute. Here are their numbers. The contents are solely the responsibility of the authors, me, and not necessarily represent the views of NIH and NCI. So I thank you for your attention today. I believe, Simone, we have time for questions.